Following her master's degree in data science, she completed her PhD in computational and systems biology at the Institute of Cancer Research in London. Heba is now a Sir Henry Wellcome Fellow at the Institute of Biomedical Engineering and the Big Data Institute at the University of Oxford. And she'll tell us today about her recent work in the really important field of developing tools for analyzing very large data sets and visualizing all that data that is gained from there. Please, Heba, go ahead with your presentation. Thank you.
Thank you, Johanna. So thank you for the invitation, and thank you everyone for tuning in. So I'm based at the University of Oxford, and I am interested in studying tissue architecture and how it changed in cancer. However, today I will focus more on the technical aspects of my work, so on analyzing and visualizing imaging data. So as you appreciate, I think this audience know that there has been tremendous advances in imaging technologies recently, in terms of from high resolution to tissue imaging or imaging of 3D models. All these aspects is generating a huge amount of imaging data. And it's the main thing for us to be able to learn from these fantastic data sets is to be able to quantify it in order to enable statistical analysis or applying machine learning to understand the signals and the information in the images. And luckily, in the recent years, there has been good progress or excellent progress in the image analysis community, and we have a very good growing community. So in terms of methods for detecting objects in the images or segmentation of various events, and of course, deep learning has been very successfully applied in these areas. And once we have quantification or measurements of the images, always the hope that we will discover new biological knowledge. However, when we work with imaging data, it's quite challenging to go from the measurements to the biological knowledge. So the image analysis is like the tip of the iceberg, and actually there is a lot of behind dealing with how we can analyze the images, like identify the phenotypes, handling the heterogeneity, or visualize the data to link quantitative phenotypes to biological processes. And I spend a lot of my time doing the image analysis, but actually larger amount of time just trying to analyze the data and looking at different ways we can get the most of the information in these images in order to discover a new biological knowledge. But today I will focus on one aspect, on data visualization. So I think if you think carefully about it, we use data visualization a lot, like every step of our scientific process probably involve some data representation. So from acquiring the samples and looking at the quality, or trying to see what features you're gonna develop, or transforming the data to make it sure it's normalized or z-scored, or just looking at the relationship between variables and trying to understand if there is an interesting hypothesis or interesting differences. With data modeling, like applying clustering or classification as well, and all the way to representing complex concepts to understand it better. All these aspects, we use data visualization in order to understand our data better. However, somehow we don't get so much training in data visualization, and actually there is a science behind how it works. So I will try to give a brief introduction on different aspects of data visualization today, and bring you a talk about some of the tools I developed. So yeah, as I said, I will give some brief introduction, but also give some brief use cases, looking at different imaging data, how we used image analysis to quantify it, and then how we can visualize them. So let's start from the basics. So data visualization is the presentation of the data in a pictorial or a graphical format. So for example, what we mean is like we map the numerical data to a graphical element. And here, for example, in a bar chart, we're mapping the data to the length of the bar. And here the length of the bar is not absolute, it's just relative in order for us to see the information. And why this is important is because a human brain processes information using charts or graph in much more efficient way and can detect patterns much easier than looking at just pure spreadsheets or numbers. So just to give you some motivation why its data visualization is so important, I would like to present you with this example that is called Anscombe's quartet data. So Anscombe is a statistician who came up with these 4 data sets. Each data set has 2 variables, X and Y, and have 11 observations. And if we look at the statistics of these data, we see that they have very similar mean and variance, and the correlation between X and Y is very similar. If we try to fit a regression line, we also see that they are have similar regression line. So we might infer from this that actually these data sets are very similar and maybe capturing similar phenomena. However, if we visualize these data, we see completely different story. So we see that these data sets have very different structure. And this is much easier for us to see when we plot the data. So this is show why it's so important to visualize and represent the data in order to see what is the structure and use the human vision in order to identify any problems. Another aspect that we can also so important to think about is that data visualization play an essential role in science communication. So especially when you're talking to, we talk to lay audience or decision maker, sometimes it's not only presenting the statistics, but also presenting it in a way that attracts attention. So for example, this is a popular example from Florence Nightingale, nurse in the British Army, and she observed that there is more deaths from disease than from wounds in the battle. So she collected the statistics over several months, and what you can see that she chose to represent her data in a more interesting way than just going to the queen or the decision maker and saying, oh look, this the statistics. So she drew this graphical representation by the length of each section here is representing the number, the percentage of deaths. And this is give a very clear message that the deaths from disease is actually much more than from the wounds, which call for some changes in how things are done. And by this visualization and by communicating this with the queen, she was able to bring reforms to the army and convince decision maker that they should do something about it. So what this graph aims to for Nightingale was affect through the eyes what we fail to convey to the public through their word-proof ears. So having very compelling visualization can be really a way to change people's mind about things. So when we think about our science, it's good always to think about how we can present it in a way that engage people that might not be very interested in it. So just to introduce you to, okay, visualization is as a field, what we think about is visual encoding. So how we can map the numbers to graphical forms. And the bar chart is one of the things, so the length of the bar. But the other things is the position, so in scatter plot. The position of points in X and Y is another way to encode the numbers. Other ways is like the area of shape or if you have a categorical variable, you can use different shapes or different textures, as well as the orientation. Color is also a popular way to encode variables, especially with heat maps. And heat maps basically we encode the variables to a gradient of color or a range of colors. And you can see here the higher values are darker, the lower values are lighter, and here just data, some shape features for different breast cancer cell lines. And what is nice about heat maps is that you can represent multiple high-dimensional data very efficiently, and they are very popular in bioimaging and other fields. Another way is glyph-based visualization. And with glyph-based, the idea is we can combine different visual encodings in a way that, like you design the visual encoding, you can use different things like the position, the shape, the angles, etc. And here, like one popular form of glyph-based visualizations called the star glyphs. So for example, here the 9 shape features, different cell lines. And we have the length of the bar, each bar represents a variable value. So this is nice. It allows us looking at different variables simultaneously and see the difference, because when one of them changes, the shape of the star is just very different. However, it's difficult to interpret this because if you don't put the variable name next to each one, it's very difficult to keep relating which variable is which bar. So this brings me to what we call visualization effectiveness. So in visualization effectiveness, what we ask is, there is different aspects. What is the purpose of the visualization? Is it mostly like trying to have an intuitive way to look at the data, or we want to the most accurate way to communicate the data, or we want to bring attention? Of course, we shouldn't compromise any of these, like try to achieve all of them. But sometimes there is elements that are more suited for one of these, while the other aspects have lower weight. So in terms of accuracy, there has been studies that actually, even if the graphical element accurately represent the data, how the humans see it is a different story. So this is the complexity of data visualization. There is a psychological element, or how the perception of our perception work. So what they found that actually, when we use the position and length of bars are the most accurate. But if we use something like the area of the object, is not as accurate. So we, as a human, we can vary a lot how well we judge this information. Interestingly, the color is one of the worst ways to represent data, because we are really there is many problems, but also we can vary a lot of how we see colors. So here, one of the things that actually how we see color is affected by what is the context, what is around it. So here, an example, these 2 boxes, if you look at them, considering their surrounding, you might think they are different. But if you put them next to each other, they're actually very similar. And here, another example where we have these boxes that maybe have different same color. However, if we put them next to each other, they're actually different. And here, another example in a heat map where we have these 2 boxes that look different, because this one look higher because the surrounding is lighter, and this one might look lower because the surrounding is darker, but they actually the same exact value. So it's good to bear in mind that actually, when using heat maps, they are useful, but looking at the numbers maybe also can be a way to ensure that actually you are interpreting the data in the right way. So next I will talk about other ways to represent the data. So my own I found it like in my own use of data visualization that I needed different way to represent the data, and I invented new tools to represent imaging data. So I will talk how this started with PhenoPlot and a use case looking at the breast cancer cell line data. So just to give you a brief overview of the data, we have, this is a plate view of different cancer cell lines, and 18 of them are breast cancer cell lines. So each image is basically representing an image, a field of view from well. And we're interested to see what is the difference between the different breast cancer cell lines in terms of their morphology. So I worked on this use case during my PhD at the Institute of Cancer Research. And as you know, with the imaging, we need to come with automated ways to analyze the data. So we segment the nucleus, we segment the cell, and then we were interested in epithelial to mesenchymal transition. So I developed features to capture the changes in, or that capture things that can reflect motility. So for example, here I'm segmenting the protrusions, and the protrusions are extension of the cell body that allow the cell to move or to grow, like it's like having a foot. You also observe like this indentation at the edge of the cell. They are called ruffles, and we can capture them based on the variation in the intensity. And they are also another process that the cell used to explore its surrounding or sense its surrounding, and also involved in motility. Another aspect is the context of the cell. So here, what we're calling neighbor fraction is the fraction of the cell border that is in contact with other cells. And also we can capture whether the cell in colony or not. So from these measurements, then I averaged this data for each cell line, and I clustered them to see how they are different or similar. And what we have is basically, as I said, I selected 9 morphological features, including the cell contacts, the protrusions, the dimension of the nucleus and the cell, and the roughness, as well as nuclear and cellular texture, which reflect the variation in the intensity in the cell or the nucleus. And looking at this presentation, like representation, I see that actually cluster 5 have high nuclear and cellular texture, also cluster 2 a bit, but cluster 2 on the other hand have high cell adhesion, and cluster 1 the same. And for me, because I was working on continuously on these data sets and trying to make sense of what are the differences between the conditions, it was really difficult to every time understand what these measurements represent, or what is the phenotype. And of course, we can go back to the images, but because these are based on hundreds of cells, it's quite difficult to try every time go through many images and understand actually what is the average cell or what our numbers are capturing. So especially like if there is a lot of heterogeneity, like you see here, cluster 4, we have some very large cells, some small cells. So this is like a challenge that I was dealing with, and I was sometimes drawing by myself, okay, this is big and this is high cell adhesion, etc., like just trying to make a picture of the things that I'm seeing. And the same thing, if we use bar chart, it's maybe accurate, but it's really difficult when we have multiple dimensions. It's really difficult still to understand the data, especially that you need to go back and forth to see what each feature is representing. So what I came up is completely new way to represent this data. So based on the numbers, to draw cartoons or draw glyphs that look like what we are measuring. So I used the glyph-based visualization. So I designed various new visual encodings that when we put it together, it looks like what we are measuring. So you see like representing the cell as an ellipse, and the nucleus is another ellipse, the irregularity in the border as a jagged line, and cell adhesion maybe as a thick line as at the border. So what this helps us then is when we have the numbers, we can plot them in a way that help us understand the data much better and relate it back easily to what we measured. And also another advantage of this approach that we can capture like 10 dimensions simultaneously easily because everything is encoded in different elements in these shapes. So here some of the, just breaking down these visual encoding elements, and many of them I designed like they were new encodings. So for example, the dimensions can be mapped to the ellipse dimensions. How much the border of the ellipse is covered by a red line or a thick line can reflect, for example, cell-to-cell adhesion. It's the same thing, but we can do covering the ellipse border with symbols. So you can use dashes, but we can use stars, like you can choose the symbol shape as well. Also, I designed something called proportional filling. So how much the variable value we fill the ellipse with symbols depending on that. So this is go from low to high. So you can see how it is with the variable value, it's go higher. Sorry, so this is can represent the number of organelles or number of specific, like abundance of specific protein. We can also use the color. So we still love colors, despite they are not very accurate. So we can use the gradient of specific gradient to represent protein levels or abundance of certain structures. Another element is the spikes. So again, how much it go around the ellipse represent how much, for example, irregular it is. And here, for example, I use it to represent roughness. Other elements that were in PhenoPlot, also I used the protrusion, so half ellipse and at the top, and how much the height of this half ellipse represent the protrusion area. And then the jagged line will be mapped to that border rather than the ellipse border. So now coming back to this data, I map these measurements, the average of each cluster to these measurements. And like here, the dimension of the cell, the dimensions of the nucleus, the protrusion, the roughness, the neighbor fraction, but also the texture represented as X symbols in the main ellipse and yellow dots in the sub-ellipse. So now plotting this data in PhenoPlot, what is give me a pictorial representation of these numbers that I have. And this representation is much easier for me to understand because it's straight away I see these, okay, small cells while these are large. And while this one is very roughly and have not low cell-to-cell adhesion or neighbor fraction, this one have higher cell adhesion and less roughness. And why this is easier for me to understand is because the elements I designed look like what we measured. So I'm helping the brain, like reducing the burden on the brain to make all these calculations and build the picture. It's just like now it's easier to see, okay, I know this is cell adhesion. I know this is irregularity because of how it looks. So it's like the similarity of what we're measuring to the visual encoding is what helping me to interpret the data better. And what's interesting with this representation that we are not using the position of the glyph. So we still have a room to use these things. So here, these cell lines, the same cell lines in principal component space. And you see, it is a good way to see the difference based on all the features, how they are similar or different from each other, but it's really difficult to interpret why they are different. So what we can do is to place the glyphs we have for each cell line on the position. So it helps us to see this information all together. So here now with these PhenoPlots or glyphs, we are able to sort the data in a way based on the principal component space or dimensions. And what we see here that actually these cell lines have mesenchymal-like morphology, so they are elongated, they are very roughly, have low cell-to-cell adhesion, while these cell lines are more epithelial-like, so they have high cell-to-cell adhesion, not much protrusion, and not much roughness. We see also some cell lines that seems to be in the middle, and this is of course known in cancer that the cells go through epithelial to mesenchymal transition. So basically this also can tell us what is the transformation happening in these cells. So that was very good, and we were very successful with this. But as someone who worked always with imaging, I had more ideas to how we can improve this. So the first design was, okay, we have one main ellipse, one sub ellipse. We can't arrange them. We can't change much about it, and there is some mapping to each of these shapes. It was also in MATLAB, which meant that the accessibility for wider audience maybe was more restricted, especially that it's commercial software. And I was also thinking about always different visual elements that maybe represent other aspects that I didn't measure in my data. So this is where Shapography came. So it is building on PhenoPlot, but it's giving us much more flexibility. So first thing, it's web-based app now, so it's much easier to use, and everyone can load their data and just start playing with it. And the main thing is also that you can use various shapes. So we have basic set of shapes, but also you can the user can draw their own shapes if they are if their cells look a bit different from like a square or a circle, or they're representing more like organisms like zebrafish or organs like liver. So it's giving the user flexibility to draw a path that then they will map the information to it. But then what is really what I'm realizing with working with this is actually that we give the user the ability to combine different shapes. And we're still working on the possibilities we can do with this, but the basic ones is, okay, we can combine a cell ellipse with nucleus ellipse, or we can draw a custom shape for the cell, but we can also represent a well and the wound, or start representing more complex things, because you can design depending on what you are measuring, the different visual encodings that you want to map your data, and this gives us a lot of flexibility. And you can check the website, but just very briefly, what the user need to do is to load their data, and then maybe you will not see much happening when you load your data, but basically you can then add shapes, and to draw a custom shape, you can use this one, this icon, and then you will have the objects, what you added in this right panel. You can rename the object, and for each object, then you have all the possibilities, different visual encodings that you can map to the these shapes. So for example, the length and the width. If there is time, I might give you like a brief way, a brief demo of how this works. So then, like, just here another example, like I have data from wound healing and experiments where we have like cells ablated, and then a artificial scratch wound has been induced. And over 24 hours, we measuring how much the wound has closed. We have this under different gene knockdowns to try to observe, to see which gene knockdowns affect cell migration. And the challenge with this data, it was acquired at 4x, so because it was high throughput screen. And the main thing the biologists looked at is the wound area, but just looking at these two images, you can also see that the organization patterns of these cells is a bit different, even if it is like low resolution. And the main challenge for using this data to detect single cells, it was the a lot of heterogeneity. So somehow in the experiments, we see some cells that are with nuclear stain, some with membrane stain, and some of them with combination of both. So what we've done is we developed a deep learning approach to detect cells in these images. So taking an input image, we train a neural network to predict for each cell what is the probability that at each pixel there is a cell or not. And what this allowed us to have a robust detector of cells based on different range of markers. So it doesn't matter what the marker you have as an input, we're still able to predict the cell's localization. So here you have an idea how the localization works. So even when it is low signal to noise ratio or the membrane stain is very faint, we are still very reasonably well detecting the cells and localizing them. So from this, I apply this to the wound healing data. And from that, what then I done is apply Voronoi tessellation, which allowed to detect assign polygons to cells based on the pixels that are nearest to the center of the cell. And what this allowed us effectively to do is to measure how many neighbors and the local cell density for each cell. So what you see here is basically now taking the control and two gene knockdowns that if we look only at the change in the wound area, they will look very similar. They both reduce the wound area. However, when we consider local cell density, we see that they have different effects. And this is important because when we think about cell migration, it's important to consider different aspects, not only the change in one dimension. And by looking at single cell localization, we can capture more on what potential mechanisms have been altered. So now just like visualizing this data using Shapography it is a very simple data set. We only have two variables. So we have the change in the wound and change in cell density. And if we look at this data, there is you can appreciate because of the differences between the initial wound and area. This can be difficult by the eye to look at what are the changes happening. So what I've done here is to add a square to represent the well, while the wound area, the wound height is represented based on the height of this rectangle. And the color and the surrounding of the well is basically representing the cell density. So the higher it is higher number of cells, the lower is lower number of cells. Also, we can represent the same information using the density of the mesh. So here, darker color, the more dense grid means that there is more cells, and lower density indicate less cells. So what you can see here basically is actually AKT2 reduced wound closure. But if you look at the cell density, it is low. So there is much less cells than the MOC. While BLCG1 have similar wound closure to AKT2, but have much more cells, which means that actually these cells have maybe affected the cell migration much more than AKT knockdown. And if we look at this picture, it's much more difficult to make sense of all this information, because it's still you need to think about it a bit more than when you look at the picture that's saying, okay, this is more easily linked to what we are measuring. So another use case and more complex use case is using multiplexed imaging data, which also is very popular now. So it's good, like we are thinking always how we can try to capture this information. Especially that when you have 40 or 30 markers, even overlaying on top of each other, it is not possible. And still, like we're still working on it, but I'm gonna present the use case, and it will be good if anybody has more ideas. So basically what we have here is a study from Lucas Belkman's lab where they developed this cyclic immunofluorescence. So they stain 3 markers and wash and stain again. And they have 40 markers captured in HeLa cells. Sorry. So markers include things measuring transcription or early endosomes, EEA1, or mTOR signaling. We have nuclear membrane protein, etc. So we have 2,000 HeLa cells from, like I obtained it from the publication, and cluster these cells and plot them using UMAP. And UMAP is very nice to get the overall structure of the data based on 40 dimensions. So you can see that there is some structure, there is 60 clusters here. So if I take the average of each cluster, sorry, and then try to look at all these measurements, it is doable. We can look one by one, but it's still not very intuitive and easy to interpret all this information. So using Shapography, I picked like around 10 variables that I mapped to like different objects. So here I have draw a cell like this to represent the data, and then added a square to represent the cell context. And to represent different protein data on the border, I just added multiple layers. So copy the object and layer them together. Added an ellipse or a circle to the nucleus, another circle can be any organelle, but you can mitochondria or centrosomes. Now, once you have the object, the structure, and of course, when you draw these things in Shapography, you can change the position, you can change which one is on the top, which one is on the left. So all these are, you flexibly can design all these encodings. Now, what we can do is to map the data, the variables to these different objects. So for example, we can map one protein level at the periphery as amount of symbols going around the cell, the density of the grid around the cell represent the local cell density. This this another cell layer can represent another protein signaling at the periphery, and similar here. The endosomes can be represented as X symbols filling the cytosol. And similar for the nucleus, we can represent multiple features. For example, here the abundance of certain protein in the nucleus, abundance of protein on the nuclear membrane, and etc. So this is basically, once you have the structures, you can map you can choose which features, which variables to map to these objects. So here, what is like, it's not basically very relevant, but it's what you have, what I chose to represent, for example, mTOR signaling to the cell periphery, and here like cell proliferation, BCNA, and here nuclear pore protein to the membrane, while the abundance of different like mitochondria is the color of this organelle. So here I'm abstracting all these objects. And the centrosome is the color of this object, while the transcription based on YAP is on the color of the nucleus. So even the nucleus now have 3 variables mapped to it. So now I plotted these clusters on the position on the UMAP, and this will make it much easier for me to look at the data in a more intuitive way and try to make sense what these clusters based on UMAP mean. Does they make sense? Are they biologically relevant? So I can see, for example, cluster 4, 6, and 2 have much higher, say, local cell density based on the grid density. And we see that here have much higher mTOR signaling, while here much more S6 protein phosphorylation, while this cluster 5 have higher YAB levels. So this is a Pictorial representation that can assist. We can plot it next to a heat map and still like help us to understand the data and look at different dimensions simultaneously in a way that help us interpret what clusters identified based on UMaps or other approach means. So of course, you don't need always to create everything from scratch. So we provided some templates on the website that you can start just mapping your data to. We have the demos, and also the demos, it can be edited, or you can change the mapping of the variables. And whatever you create in the website, you can save it and you can upload it later for later use. So you don't need to every time change, start from scratch. So I'm just maybe briefly showing the website. Can you see the website?
Yes, we can.
Okay. So basically here we have different examples, different data sets. So for example, this is the multiplexed imaging example. And here I named the objects, for example, but what is nice is you can hide, like while you're working, exploring with your data, you can hide an object and think, okay, this is too much information, let's look at one thing at a time. Or you can also change, for each of these objects, once you select it, you see which variables are mapped. So for example, here in this, we have CMYC, or the map to the color, this gradient. We have fill symbols is mapped to the BCNA protein, which is basically these dots in the nucleus. And you can change the settings. It's very customizable, so to suit all tastes and abilities. Border symbol is the symbols here on the border of this object. And here also, like the templates, we have like, we defined a few templates, but we're looking forward people to apply to their data. And if you have like another use case, maybe you didn't think about, we will be interested to hear from you. So you can just contact us or submit your creation. And of course, you can save as PNG or SVG, so you can edit it in Adobe Illustrator, etc. And the format of the data is very simple. So just CSV file. The first column have the object, like the sample's name. So here we have different clusters, for example, and we have different variable names. So here we have a breast cancer tumors. So we have from Metabreak dataset 2,000 tumors that were clustered based on the genomics, and they will have some phenotypic data. So we have the size of the tumor, how much lymph nodes the tumor has spread to, the grade, and the cellularity. So I can show you very quickly, like you can upload this data set by going to load data and the breast cancer tumors, and then create an object. So for each cluster, we added a circle. For this object, you can rename it by clicking on the pen and call it, for example, a tumor. And then you can change different aspects. This will change globally, so it is not data mapping. But for today, I'm just showing like the data mapping. For example, we can map the tumor size to the length and the width of the object. So already the information now mapped to the dimensions mapped to the circle or the shape that you created. The gradient can be used to represent the grade. So the grade, if it is lower, it means less invasive or less critical for the patient, while higher, it means worse outcome for the patient. The fill symbols, like you can imagine the tumor filled with cells. what cellularity basically measure the density of cells, and we can represent it as symbols. You can change the size as well, and if you want the color. And for each of these elements, you can just click on it to expand it, click on it to collapse it. Another feature we have in this data set is the lymph node. So we can represent it as something on the border because spreading on the border of the tumor. So lymph node, and we have different symbols. We have dashes, a star, but it's just depending on what you are measuring or what you think it's most appropriate for the data. So, and of course, sometimes it's about design, so you can hide something and put it back again and test the combinations you want with your data. There is aspect data normalization. So what we do is normalize the data between 0 and 1. It's done automatically if your data is not normalized. And the positional mapping is basically what allow you to plot on a scatter plot. So now I can organize the data on a scatter plot, and basically now I have the clusters I showed you in like a pictorial representation where we see these clusters that are based on the genetics are more invasive and more cellularity for cluster 5 and 9, while this seem have lower grade, lower cellularity, but it is larger tumors. While this one is maybe smaller tumor and not much spread to lymph nodes, but has high cellularity and high grade. So it's just a different way to represent the data that can be more intuitive to understand and to link to the variables that are measured. Of course, you can see the table here, and you can also look at a heat map of this data to look at the values. Currently, I find it easier to work with smaller number of points. So if you summarize your data in clusters or have some classes rather than trying to plot 100 things, it works with 100 things, but because there is so many information in each glyph, sometimes the human brain can maybe deal with so many like 100 points at one time. And yeah, once you're done, you can save your template so you can use it later. So you have the same mapping, just load a data set with the same column names and you will have the same representation. So I think I will summarize here so we don't run out and we keep time for questions. And so in summary, Shapography allows effective way for data visualization to facilitate the interpretation of phenotypic data. It's very flexible because the user can create the object shapes they need and combine different objects to represent the different things they are measuring in their data. And maybe for some audience, for example, showing cell images can be very difficult for lay audience. And I know that because I started my PhD as a computational person, and initially looking at cell image data I don't understand what is that. So sometimes having this abstract representation may be better to communicate your data with the audience and maybe attract more attention because of the glyph nature of it. And of course, there is still things we need to work on. So as I said, when we have large samples, and the other thing that maybe that different variables can interact with each other. So there is always studies in human perception, how these different encoding, when we create it, how they can impact each other. And I think it is over practice, once we define a standard like templates, the user could use to it, and we can use it like in general as a standard, which is the case in many fields. Like we define some visualization, and by the time we get used to it and interpreting it more effectively. So yeah, at the end, I would like to thank my funders, the Wellcome Trust, for funding my research, and which gave me a lot of flexibility in visiting different labs and pursuing the research that with high independence. And the people who done the work, Muhammad and Johanna, and my collaborators. So The PhenoPlot was developed while I was a PhD at Chris Baker Lab. And thank you for listening. And please try the website and let us know your thought. We are looking for ways to improve it and how the users can use this website. So it will be good to hear from you. Thank you for listening.
Thank you for this great presentation. It was very interesting. I found it really, really cool.
Thank you.
Very useful tool. I think that I'm at least very excited to sort of go and play around a little bit with the sample data sets you have on there now.
Yeah, that would be great. Yeah.
Do we have any questions from the audience for Heba? You couldn't just unmute and ask your question directly if you want to. Maybe I'll give people a chance to think because of some questions, because I have a couple of questions that I wanted to ask you myself, and maybe I'll then go first and people can think of their questions. So the first thing I was wondering, what if you're thinking about how you can represent sort of variance within data sets using these tools, because sometimes that can be an important characteristic as well. And these really focus on like the median value or something. What are your thoughts on this question?
Yeah, I think that's a really good question. So we are also thinking about how we can represent the variability in the samples. I think just designing it in a way that because there are already so many elements, adding the variance of all these objects might be challenging. But I think we will have maybe in the future a better way to incorporate this as well in a way that's still intuitive with it, and the user is able to deal with this information. But this is a great question.
And we have a question in the chat from Sybil. Sybil says that she really enjoyed listening to your very clear presentation. And her question is that tumor cells are usually not that stable. And there are a lot of things that shift, like epigenetics, the microniche. And the question is whether you're planning or are able to do shapography as well, looking at using longitudinal studies, so looking at how these same cell lines change at multiple time points.
Yeah, that will be very interesting. I think shapography is not limited to a specific data, is basically what you have. So if somebody have the detailed data, we will be happy to chat with them and see how we can, maybe if there is features that we can add. To be honest, I don't have currently longitudinal data. Maybe I will look into live imaging. That's what we're thinking. But of course, like it's at the end, if you have your data in Excel file, you can export them as a CSV and load them in the tool. So it doesn't matter if it is longitudinal data or just mean data or like even other things. So it is basically, it's generalizable tool that you can load any data set to it.
Great. Are there any more questions? Yeah, go ahead.
I was interested in the investigation of breast cancer cell lines. And my question is that sometimes you get these clusters and the same line belong to the cluster 1 and cluster 5. Maybe I missed them, how it is possible, but how you deal with that one, that sometimes it's cluster 1, cluster 5, or the same cell line is in this. You can find it in both clusters.
You mean my analysis?
Yeah, it is what you showed it. Like I remember it, like MCF7 was in cluster 5 and cluster 1. Even GIMP cell lines, both clusters. I thought that one can belong only one. Maybe I'm wrong.
No, maybe, I don't know. I will check the slides, but it should be one in one. It might be a typo, but I think I will check yeah, it should be one, yeah, in each cluster. It shouldn't belong to more than one cluster.
Oh, I see. Okay, but okay, maybe you can check your slides.
Yeah.
Maybe I was wrong.
There is MCF7 and there is MCF10, so 10A. So
MCF7 and the GIMP 1.
Yeah.
I focused on that one because I worked with the GIMP. Actually, we started the GIMP one with the Finnish operator, and that's a resistant cell line. It's interesting cell line.
Yeah, I see it on one cluster, but yeah, I think I can't share my screen. this one, you mean like MCF7 is this cluster?
Yeah, but then you had the circles, and then they showed they belong to two circles later on.
The glyphs, you mean? Here, you mean this one?
I don't know. I can't see it.
Is this maybe this? Is this one or?
Yeah, you see that? Cluster 1, cluster 5. Gene 1. This is all.
Oh, this is a type, but this should be MCF10As, I think. Okay. No, actually, this is a type. Oh, okay. Thank you. Very good spotting. Okay.
Or maybe this is type with the same row. Yeah. Yeah.
Yeah, I think it is. Yeah, it's.
Now I can see it. It's a typo.
Yeah, it's just my mistake. Thank you for
paying attention. You see that? Okay. It was a nice presentation.
Good.
Anyway.
Good. I will correct this.
Great. Do we have any more questions? I think Sybil posted some other suggestions into the chat. Sybil, do you want to just unmute and talk about this yourself?
Yes, of course I can. I hope you can listen to me. There's some noise in the background, but I hope it's still clear.
Yes, we can hear you perfectly.
Thanks again for this clear talk. I really liked it. I really enjoyed it. I'm a vet. I'm veterinarian. I'm working in Vienna. And I just wanted to add or suggest that you add functional data to your Shapography, because you keep to morphology, which is good. It's a start. But my suggestion were to add estrogen or other hormone levels, because we do not observe breast cancer nor breast cancer mets in female dogs anymore, since we have the regimen to castrate them quite early in their maturation. So, well, you can't castrate females, of course, but of women, but I think there's a close relationship between incidence of breast tumor and hormone levels.
Yeah, that's a great suggestion. Actually, we have, I think, this data. So it's just now we can add an object and represent it, the color of that object surrounding the tumor to show the level of hormones, or in another way. So I think it's very easy to map this additional information. I just didn't happen to think about it, but you can represent the hormone level as like symbols on the border or as a color of the shape. So it's quite, you can actually map any data type. But I think in my case, we were looking at the morphology, but even for those cell lines, we know the ER, PR status. So maybe I will update the template with this for other people to also appreciate that they can also look at this information. Thank you for the suggestion.
Okay, well, I think with that, we're going to wrap up the Q&A session. Thank you, Heba, for answering all our questions.